Chapter, and we just provide a venue to talk about new tools, technologies, methodologies. Sometimes they're new, sometimes they're in development still, or they could have been around for a long time, but just used in novel ways or interesting ways that someone wants to share with various attendees. So today we have Becky Steck from the Kretzler Lab in Nephrology and Ross Patterson from also sort of the Kretzler Lab and the Bioinformatics Core. And they're going to talk to us about Nephroseq. And those of you who have been around for a while, this was formerly Nephromine.
Do I stand for this? Do I sit? It's off. Where do I go? Okay. I'm going to do the half and half then. I'm going to be right in your way. How about this? All right. Before I get started, I have a quick question for the room just to help me calibrate how well this is going to go. Any Game of Thrones fans out there? A couple? Okay. I am a Game of Thrones fan, and so I may have incorporated Game of Thrones to every slide accidentally. So just to let you know that's coming. And if you're not, I will try my best, as is Winter. Yes, I will try my best to explain the references where applicable. But as Marcy said, my name is Becky Steck. I am actually a software project manager in Dr. Kretzler's lab. There's two main bits of software that I work with. Nephroseq is one, which we're talking about today, and Transmart, which I work with Zach on. So hopefully you guys have had a Tools and Tech on Transmart already or know about it. Otherwise, okay, I'll be coming back. But with that, I will kind of say what the agenda is. We're going to just kind of give a quick overview about what Nephroseq is in general. And then we'll go into a demo about it too at that point. Afterwards, after the demo, we'll talk a little bit about what some challenges are that we are experiencing right now. Hopefully get some ideas from you guys about how to take over them. Tell you a little bit more about the team that's working on it right now and have some time for questions. Yes, Rich?
You're going to do a live demo?
Live demo.
Okay.
So I'm going to need someone to volunteer to write down the bugs we identify, because there's nothing like a live demo to bring out the bugs. So as long as we got, okay, Ross is volunteering for that. Excellent. So that's what we're going to do today. All right, so what is it? What is Nephroseq? Nephroseq. You might know if you've used Nephromine in the past, but we like to think of this as a lot of words because we want to get a nice like buzzword bingo vibe going on. So it's a web-based app. Okay, so you can go to the web to use it. It's a data repository plus search engine plus analyses. So in essence, we throw a lot of data in there, and we've got some pre-computed analyses that you can then use and visualize data in a lot of different ways. This is gene expression data that we're talking about, and it's specifically for renal diseases, kidney diseases, okay? So a lot, like the main use case here is to look at differentially expressed genes. You can also look at co-expression, outlier expression. And one of the things that we try to make it is something that you don't have to be a bioinformatics pro to use. So in some cases, we're hoping the interface is intuitive, and maybe in some cases we don't go to the depth that a bioinformatics person might want to see it in, because we are trying to make kind of broad strokes relationships between different data sets. And so it's less important that we have every bit of specific minutia for a data set right, as more that we've made it so it can be comparable across data sets, still keeping its important properties, right? So one of the ways we do that, for instance, is mapping all these genes to, or the data sets to the same gene model. And there are pros and cons to that, but for the purpose of this application, that's the right way to do it so that you can actually make comparisons across different data sets. Another thing we've done is taken the different metadata, so data about the samples, maybe their the demographic information about the patient sample, and we've developed our own internal, almost an ontology, but that's a little too sexy for what it is. I'll call it maybe more just a hierarchy of terms that we've organized and that we've developed ourselves. And that way we can kind of map every data set into that same, round peg, square hole situation, so that, again, we can have these analyses be comparable across different data sets. And the other thing we really like about this is we're hoping that it encourages knowledge sharing. So you can, bookmark your things. You can then send them around to other people. We're not trying to hide a lot here. It's really easy to get access if you're a researcher, and we're hoping that this will facilitate lots of new discoveries. As a matter of fact, just an aside about the Kretzler lab, Dr. Kretzler has kicked off this year he's called it RPC2, which stands for Renal Pre-Competitive Consortium, where he has 3 different pharma companies that are all working together with the same data to try to come up with new targets, in essence, to come up with drugs that can help cure different renal diseases. And this is unprecedented that these three competitors are working together just to try to make this go faster. And one of the tools they're using is Nephroseq. So it's kind of cool. But it's also really helpful, especially with this particular audience, to talk a little bit about where Nephroseq came from. So Ross pointed out as we were walking in the hall here, if you've heard the name Dan Rhodes, he's part of the first class of bioinformatics master's students here at the university, and he started something called Oncomine back in 2006. Oh, and sorry for the non-Game of Thrones things. People, this is kind of a spoiler, so but it's just a fan theory. It's not been proven yet. Wait till season 6, okay? But so Oncomine was launched in 2006, and it has many of the same aims, right? But this time it was for cancer. When Dan started, he actually spun out of the U and made his own company called Compendia Bioscience. And as it happens, Ross, Rachel, myself, and Chris Gates were all worked at Compendia as well. So we're intimately familiar with Oncomine too. So what happened is he wasn't sure at the time if the powerful thing he had is software that could help with cancer, and that maybe he was really good at coming up with software to help with cancer, or maybe it was just really good software that could help in other diseases. So at the beginning, the early days of Compendia, we kind of split and went in two different directions and said, well, we'll get Oncomine going, but it turns out there's this guy at the U, Dr. Matthias Kretzler, who is interested in the same thing. He's willing to give us some grant money to do something called Nephromine, which is the same idea, in essence, the same application, but with a couple differences so that we could use it for nephrology. So at that time, Nephromine was born. In the intervening years, which there are a lot of them, almost 10, Compendia was purchased by Life Technologies, which was then purchased by Thermo Fisher. And as part of that, Dan, probably before that time, realized that Compendia's bread and butter was oncology. And we had, at that time, made a lot of other oncology software, but we were keeping the lights on for Nephromine because we had grant money coming in, things like that. Well, as Life Technologies and Thermo Fisher got involved, this was less important to them. They wanted the cancer software. So at that point, the university worked with Thermo, I guess, at that point, to bring Nephromine back in-house. And part of that was that we needed to rebrand it as our own thing. So that's kind of where Nephroseq came from. But what's really exciting about this is that before the university got it back, we hadn't updated the Nephromine application in 3 years. So, but we still had a really strong user base. We got about 1,000 users right now who enjoy using this tool for their research, and they would love to have seen new data and new features. And so we're really excited that now we have the ability to not only keep the lights on, but actually update this sucker. So that is the official launch of Nephroseq was January of this year. Since then, we've had a couple other app releases already, which is a big hockey stick from the 3 years prior. So, oops, this is the wrong schematic. I did not mean to throw Game of Thrones. I meant Nephroseq. Sorry, this one is another one for the non-Game of Throners. Study this, okay? It's really important. But for the Nephroseq people, this is a high-level kind of, I can gloss over this because we're going to go into each of the specific sections in more detail. But in essence, we are collecting, if you look at the top left side of that graph, we're collecting data. A lot of it comes from Geo, but other times, you know, we happen to have an inside source here where we're working with the Kretzler lab. They know everyone, and they know who's working on this type of data. They know who's publishing on it. And so we get early access sometimes to their data, and also access to just email authors and say, hey, we hear that you're writing about this. Would you be willing to put your information into Nephroseq? And not only that, but a lot of times, as part of getting the data set, a lot of Nephroseq is dependent on really good clinical metadata so that we can make these different differential expression analyses on the different phenotypes, different classes. So we work really hard to make sure that we don't just have the raw expression data, but also some data about the samples to go along with it. So a lot of times we'll write back, we'll say, hey, we looked at your table 1 where you said you collected X, Y, and Z, but you didn't provide that in Geo. Can you give it to us? Or, you didn't put that in a supplementary table. And we've had really good luck with that. A lot of times people are very excited to do that. So sometimes we're writing to the authors, but we get all that data in, and then we, as I mentioned earlier, standardize the metadata. And because of that, we can then automate differential expression analyses on that data. On the right side of the graph here is how I kind of said we mapped to a single gene model, and I'll get more to that later. At which point it all kind of goes in the same mixing bowl, and we're able to present it on the web. And here's just a couple of the examples of the visualizations that Nephroseq provides. But we'll go into some specificity here. This is a picture of the Unsullied for you non-GOTers. They are all bred to be sweet fighting machines, but they're also bred to be very similar and consistent. Not bred, right? They're not bred. They're just trained. Right. And so that's, I thought that was a nice picture for what we're doing here, because we've got lots of different data sets. And since it's tools and tech, I wanted to make sure to tell you a little bit about the technologies we use. The beginning, a lot of it is elbow grease, right? Talking to the authors, emailing them, downloading their data, looking at it, making sure it makes sense. At that point, when we put it into that hierarchy, we use Protege, which is a software that is used to manage ontologies. We just don't use a lot of the bells and whistles of it because it's what we're doing with it is pretty simple. And then we can standardize differential expression analyses, and the work to standardize those and run those diffx analyses is a combination of Bash, C, and Perl right now. So here's a little highlights right now. The data that we have in Nephroseq, we've got 26 data sets, which consists of about 2,000 samples. I think it's 11 shy. But we have over 300 differential expression analyses that we've cooked on those 2,000 samples. Some human data and some mouse data, which I'll talk a little bit more about later. Some disease, some normal. It's a really nice diffx to do, right? Compare your disease to normal samples, and lots of other data as well. And sorry, just ask me if you have a question, right? Gene annotation. This is the Dothraki Khalasar. Gosh, I don't know. I'm getting rough here. I wish there were more Game of Throners here. So this is a group of people that travel along this, what do they call the sea? It's a grass sea, something like that. Okay, I'm losing you. So it doesn't matter. It's a good picture for what our gene annotation process is right now. So we map different probe sets from the different microarray platforms that we have on these data sets. And we use the platform definitions for the most part to decide to map those to different nucleoside accessions and eventually on-trans gene IDs. So we really do only support a human model, but we have these 2 mouse data sets in there. There's a lot of mouse research that's being done in nephrology. And so what we actually did was mapped the mouse genes to the human ortholog. So it's not the entire mouse genome, but it's what we could do. And it is a little weird, but it works. And so right now we fully recognize it's a little weird, but it's working for now. So we know that's something we want to improve on in the future, but it does the trick. So we also do provide some basic information about genes in like hyperlinks throughout the application that people can pop out to other places. So, and then the application itself. So I'm about to go into the demo portion here, but just to say, this is probably a slide that Ross should talk about, but it was built in Java Web Frameworks, right? You can read just like I can. One thing that's kind of interesting is originally when this was born from Oncomine, right? I said it was kind of a copy of Oncomine. Back at the time that we were building Oncomine, we had a lot of customers from pharmaceutical companies. And a thing about working at a really big company like that, and U of M sometimes has the similar situations, is that they all needed to be, they were all on one operating system because it was a big deal for their IT departments to change to other browsers and other operating systems. So we had a bunch of users on IE6, right? It was horrible, right? But and we had certain situations, therefore, that we had to think about, knowing that these users were on IE6 and probably not going to come off for a while. So so there was a lot of decisions that we had to make to design for it to work in IE6, right, wrong, or otherwise, right? So most of the data is stored in flat files in a small relational database. And then most of these analyses are pre-computed. There's a few that happen on the fly, but that helps the app itself go pretty speedy, which I think you guys can all relate to. It takes a long time to pre-compute some of that data. So, all right, at this point, we're going to go to the demo. So I'm going to switch around a couple slides just because I want you to see the sweet slides I made. But the first thing to do is just know that the application itself, we have a .org and a .com version. Right now, they are almost identical. The only difference is the registration workflow, because we ask .org users to sign a license agreement, whereas .com users are paid users and they have signed a contract that has some of the legal jumbo in there already. So, but the idea that we broke those two apart is because maybe someday there would be, maybe our pharma customers have a proprietary data set that they want to put in there themselves, or maybe there's additional features that we would only do for the research community because they have different needs than the pharma community. So we just set that up. Right now it's easy for us to maintain those two instances, and that's kind of why we did it. So for you guys, if you wanted access to the app, you would just need to register with your UMISH IDs and you'd get free access by going to the .org version. And if you tried to go to .com, we'd be smart enough to send you over to .org instead. And then the other thing to just keep in mind as we talk about, as we do the demo, is there's really two main views that you'll see in Nephroseq. There's some exceptions to that, but one view is a summary view. So when you first dive into a search, oftentimes you'll get this giant grid box that's supposed to tell you a lot of information all at once, and you'll see one of those. And then the other view is very common, this 3-column look that I have over here, where you'll have your search options. So you'll do some searching, maybe you'll have to filter through or browse through the tree to find out what you wanted to do. And then after you select that, the middle pane will show you the results for whatever you searched for. And then the pane on the right is where you'll have the resulting visualization on whatever result you pick. So that's a very common cadence just to know about in Nephroseq. So I'll tell you that the first kind of workflow that I'm going to show you, see why I wanted to have the slides, right? How else can you get? This is Renly Baratheon. Rest in peace, Renly. Okay, so the workflow we're going to do now is if you're a student or you're a researcher who has a particular gene that you are studying and you want to learn what you can from Nephroseq about that gene. Okay, so I'm going to get out of here and go into Nephroseq and do that. Oh yeah, don't look at the next slide. Spoiler alert, spoiler alert. So here's just an example of com and org. As you can see, they don't look any different, and that is by design. Okay, so here's the live demo part. Is everyone paying attention for bugs? Okay, the first one is whether or not I can type my password in. All right, great. Never. Okay, so here we kind of see a version. This is one of those exceptions, but on the left is where we search for things. So down here, where'd my mouse go? This is where this hierarchy, this ontology that we've developed can be witnessed in all its glory. So here, for instance, in the sample filters area, you can see some of the data that we have collected, and the numbers after it are how many data sets have a value. So how many data sets do we know age of at least one of the samples? 21 of our 26 data sets, right? So a lot of this makes sense to everyone, and some of it only makes sense to kidney researchers, and therefore a lot of it doesn't make sense to me, but that is okay. So that's an example there. But in this case, in the search box, if I were to search for anything that is already in there, you see there's an autocomplete, right? And it's giving me the top 10 results. But that's an example of how sometimes my results are genes, sometimes they're analyses, sometimes there's concepts, which is another name for a gene list. So we'll get to that in a second. But first, I'm going to use a gene. I'm going to say I'm studying this gene, except for I spelled it wrong, NPHS2. Sure, that's my gene. So now that I've searched for something, what I find is that big grid I was talking about. I'm going to put this guy aside so we can see a little more of this grid. You're probably getting your bearings already, but the columns are different types of analyses, differential expression analyses we're doing. The rows are kind of data set types. So a lot of times when you write a paper, you're talking about a certain theme, right? A certain disease or a certain group of diseases. So that's loosely what you have in the rows. Now the boxes, what you're seeing there, so we'll go to this one right here, this sex and then IgAN. That's a type of nephropathy. So what this blue square is saying that 1 out of 4 analyses meet my threshold in the 3 data sets that actually have IgAN data. And so that hover may not have made any sense. We're saying that there is one analysis that has significant results for this gene, one gender analysis. And so therefore this gene is significantly underexpressed in this analysis. That's what it's blue. A red column would mean it was significantly overexpressed. And sometimes if I cruise over here, there's some over here, an outlier as well. So the the depth of the color actually means something too. If you saw the scale over there, just how significant. There's the color scale. But what I can do is click into one of these, and group is, that's actually our disease group. So I'm going to click into this. There's 7 different analyses here where it's, so now if I pop this back over for a second, just to remind you, we have a search bar, a results bar, and visualization. So what you can see is it just added 2 filters. It added the filter of analysis type of group analysis, because that's the column I was in, and data set type of CKD, chronic kidney disease, because that's the row I was in. So now it's giving me the results that where this gene is significant, okay? And so if you've used Oncomine or Nephromine in the past, this is very familiar to you, and you could probably come up here and tell me where I'm wrong. But so what you're seeing, in this case, just a nice bar chart, but you can switch over to a box and whiskers plot if you'd like as well. We show these results. Right now it's showing what we call the best reporter, but sometimes there's multiple reporters that you can toggle through when there were multiple reporters on the platform that we've mapped to that gene, right? You can also change your threshold. So maybe you say, you know what, this is a little too stringent for me. I think I just want anything with a significant p-value at all. And you'll maybe see more come through, right? So in this results pane, you see p-value, your fold change, and this here is gene rank. So this, if we had, oh yeah, go ahead, Rich.
So is that reporter a probe ID?
So great question. It depends on the platform that the data set is associated with, right? So if I scroll down here, I actually can see a little metadata about the data set itself. So this platform for this particular data set was an alternative CDF that I think we developed at the U, if I remember, for this particular data set. It was a brain array one. So so that just might be the unique ID of the brain array, like reporter identifier that we developed for that chip. But sometimes it's AFI-probe sets. So it just depends on whatever the platform is that's associated.
So there's a way to drill down and find out more information about that?
Yes, sir. Wow. It's almost like he's a plant, right? But he's not. He really isn't. But here's a little pop-up down here for reporter information that will tell you a little bit about that probe set, what we have about it. And that also varies depending on the platform, how much information we have. So in this case, there's not too much to tell you except for that's the reporter ID. And then you could pop over here and pop out to see information about the gene itself from NCBI, right? And another one is, this is that gene information as well. So here, this isn't about the reporter, but about the gene. And that's, I told you a little earlier how we throw a little metadata about a gene. That's where it goes. All right. So yes, is there any questions about that? Any other questions about that? So this workflow is something where it would be really helpful for folks to see maybe what diseases are interesting in my gene if they don't already know that, right? Maybe they can discover, oh, I had no idea that I should be paying attention to diabetic nephropathy. My lab's been focused on FSGS, good old FSGS. And so, oh, no, it's in FSGS too. That's odd. Maybe this guy's a player in all the kidney diseases, right? So there's a couple different things you could do. I'm going to skip ahead to one of the additional functionalities because that's a really good example. That one thing you can do after this is what we call a meta-analysis. Sorry, I know that phrase isn't that earth-shattering. You may have heard of a meta-analysis, right? The way we do that, though, is you can choose multiple analyses from over here, multiple results. So in this case, these happen to just be lots of different diseases versus healthy donors, right? So I'll just click a couple here. And then if you hit compare, what we're going to see is where that, sorry, this is where this gene ranks in all these different analyses. But if I remove the gene from my filter, now I have a very different view here because now I've changed from target, like from learning about my gene to now this is a totally different workflow here where I might want to learn, I might want to discover a gene, right? So in this case, we looked at a couple different analyses and said, are there certain genes, oh, my meta-analysis went away, that are, mark that down, Ross. there are certain genes that are players in all of these, or that are players in most of them, but maybe stick out like a sore thumb in one. And so what we see here is this is a meta-analysis. So now the legend, these columns are no longer samples, they're actually data sets, the differential expression analyses. These all happen to be from the same data set. But so now I can see that this gene, mTORF, happens to have been highly overexpressed in all of those analyses. So that's, maybe something that somebody might want to research and find out, why is that gene a player in all these different diseases? Maybe it's in a pathway that's just loves the kidney. And so what you're seeing here, these median rank, is what gene rank that gene was in the different differential expression analyses, right? And then you can actually see, when I mouse over, the rank it was in the particular data set, or the particular analysis, excuse me. So in the third analysis, it was ranked #1, but in this guy over here, he was ranked 299. So that's a good example of how, again, the color saturation is also trying to give you a hint about how significant. Rich.
So upregulation, downregulation are relative terms, and there's always a little bit of fuzziness about which set was it downregulated or which. So is there a way to go back to the raw data here and say, in this group I can see that it was lower expressed than in this group, to remove that uncertainty?
Yes. So it's not one click away, but in essence, what you can do is say, all right, I want to know. I'm really interested in this one, that's discovery cohort diabetic nephropathy versus healthy living donor. So I'm going to clear out my meta and just go find that one analysis and look at that. And now you've got this heat map. So it's not raw data yet. I'm not purposely skirting your question, right? But I'm working into it. So this view is a heat map, and it's just ranked by it's gene ranked by p-value. And you can continue paging through to see deeper and deeper. Now, the closest you can get to raw data, there's one thing, is just that if we got the data from Geo or something, we will link to it down here. So you could go to Geo and grab the raw data yourself. You can also export some data from within the application. So here's the export button. It gets kind of hidden sometimes. If you're in this view, it's really obvious to see, but if you're pulled your heat map over a little, it's hard. But you can export a PowerPoint of this image, which is nice if you wanted to share it with your colleagues, an SVG of this image in case you wanted to put it in a publication, or some actual data here. So the analysis gene list, gonna pop, waiting, will give you an Excel file with, do you remember how many genes we're gonna see here? 200? With, oh, there she be. Oh, this is beyond my abilities, Alex. But it's all right. You're going to see the top 200 genes. Yeah, I know. It's back there somewhere.
I can try to get it up.
It's not a big deal.
I wonder if it's actually easier than that. So down at the bottom, you got group 1 and group 2.
Oh, yeah. Is that?
So which one's group 1 and which phenotype is group 1? Which phenotype is group 2?
I see what you're asking. Healthy living donors were in group 1, diabetic nephropathy in group 2, which is usually also listed in the analysis name itself. I say usually because sometimes, like for instance, in a race analysis, usually what we do is pick one race against all others. So white versus every, black versus every. And so in those analyses, we'll just say black, and it's assumed that it's versus any other races.
Could I offer a suggestion that instead of a 1 in that column there, it says healthy living donor?
That would be a great feature, because that's a little pain, right? Yeah.
Type 1 the real phenotype.
Yeah. We can just lay, instead of just having 1 and 2 and then having to search the legend, you could just.
The reason I'm bugging you is because in our RNA-seq, people always, we give them a table that's like this, and people always say, which one's in the denominator and which one's in the numerator, and so I explained this to them.
Right. Yeah. It is fair.
I think one of the problems with that is when you have a column of 1.
Yeah.
One sample.
Oh, with a big long name.
With a big long name. What do you do that? Yeah. We could.
Spoken like a true software developer.
We could work around that problem too, but that's the other side of it.
Mouse over to get the full thing?
Yeah.
There you go.
Well, mouse over gives you legend value there, and then the actual sample as well, and the normalized expression value is the other thing you see in there. Sorry, let me go to mouse over that's higher. So
No, I think he meant mouse over the 1.
Yep.
Oh, gotcha.
If you got, you can put,
Oh, okay.
You know, that actually doesn't work. It looks like it looks clickable, but it's not. It's just highlighting class 2. But mousing over here, just one cell up and you got it. So yeah, that's a workaround though, and I like your idea. So, all right, so this talks a lot about how to do. I guess we kind of switched from target discovery or target validation to target discovery, right? But then you also got a sneak peek of exporting, and we talked about meta-analysis. The other thing to talk about is concept analysis. And concept is it's just a gene list, right? We call them concepts, you know, just to keep you on your toes, because we like coming up with our own names here, right? Whoops. So the concept is, in essence, each one of these differential expression analyses, we have pre-generated 6 gene lists from them. The 6 gene lists are the top, I'm going to drop down there, the top 1, 5, and 10% of genes that are overexpressed in that analysis and underexpressed in that analysis, right? So you may want to look at a whole set of genes instead of just one gene. Actually, this is a good time to point out that you can search for more than one gene as well. Like you're not limited to searching one gene at a time. You can throw in multiple genes and see heat maps, or toggle through if you're in that like summary view and see the different genes, right? But sometimes you have you want to see, like in that example that we were looking at where we did a meta-analysis and saw there were lots of genes that were players in multiple analyses. Maybe you want to see if there is something there and maybe figure out if there's any biology associated with it. So we have a bunch of, so as I said, we pre-compute for all these over 300 differential expression analyses. Each of them has 6 gene lists associated with them. We also have third-party concepts that we have loaded. And again, this is full disclosure because I'm in a safe space. Some of these could use an update, but and some of them came from Uncle Mine, so maybe they're not even relevant to the nephrology community. But so here's an example of some biology concepts that we have, right, and some pathway data. So maybe you have a list of genes, like you think you're onto something, you may want to see if it overlaps with biology. So the way you can do that is through concept association. So let me take that filter off where I accidentally clicked. Let's pretend, and you may not have noticed, but we flipped over to this more green theme here because as soon as I clicked a concept name, it flipped me over to the concept side instead of the data set tab. You can easily go back and forth here, right? But so I say, you know what, I'm really interested in what's going on here. I wonder if there's anything about it. Ross, why is that so ugly? See, live demo. Live demo, that's why it's so ugly. But I say I'm really interested in knowing what's going on with these genes. I want to see if they overlap with anything else in the system. And so I click whatever list I want. I'll pick 5 just for fun. So it's going to prompt me to say, okay, you probably just found the analysis you were looking for. Do you want to keep all of your results restricted to those filters or remove them? And so usually you want to remove all your filters, but sometimes you don't. So now you're going to recognize a familiar looking screen to you. The first thing we're going to see is another one of those grids, right? But this grid, instead of these numbers now are the number of concepts or gene lists that overlap with the concept you had. So a lot of times you'll see a lot more numbers that show up in this overview, which we call the concept summary, than in the gene one. So now you can see there's lots of overlap between the 5% of genes that were overexpressed in diabetic nephropathy versus healthy patients, and with plenty of other concepts. So you might be like, wow, what do we got here? I'm really interested, hopefully, right? So you could, at this point, pick one of these cells to drill down in, and remember that will add those as filters to say, okay, you were only interested in race analyses and only interested in underexpression. But maybe you don't actually want to limit that. Maybe you just want to see what is the highest association. You can just switch over your view to the concept view, and now it's going to show you the results in this panel, and it's going to sort them by p-value. So here is the concept that was most highly associated, so had the most overlap, happened to be from the same data set, but in this case it's the disease arterial hypertension compared to healthy living donors. So now your visualization screen, and again, I'm going to slide this over, just make things slightly prettier, is going to show you information about the concept that we said was that the one that we wanted to compare everything to. So here's the name of the concept, what it is. This one came from a Nephroseq analysis, right? But you can also pick those third-party concepts, those biology concepts to be, your primaries. You can click into this and see the actual genes that you selected in there, right? And then now you're seeing
left column.
Left column over here.
And scroll down a bit in there. I want to see if there's
Sorry.
anything outside of JUCKD.
Yeah.
I'm wondering if there's a systematic bias in the data.
There definitely can be. Yeah. But you will see there are, sorry, I got to get to this area. You'll see that there's other data sets besides Jew. That is the difficulty with right now only having 26 data sets. You're going to see a lot of familiar faces. But now we have some other data sets coming through, right, that still have pretty nice p-values, very nice p-values, that aren't from Jew.
Yeah, well, all the top ones were the...
Yeah, absolutely. Which, you're right, it makes sense, right? And I'm curious even if we were to get to a point, Oncomine, when I left working Compendia, Oncomine had almost 1,000 data sets. Kidney disease probably never going to have that many, right? But if there's a point when it reaches a critical mass where we maybe don't show the associations with your own data set because that's biased, right? So there are some other ones. This is actually a different data set, hard to tell, but different data set. But most of the ones at the top there were definitely the same data set. So going back here, what you see now, this is just information about that first association that we had, or whichever one I'm highlighted. And then here you're listing out the actual overlap. So here's the genes that overlapped, right? With their statistics. A couple cool things, you can export that list if you want. You can also export edges and nodes. And then from here, another neat thing is to click this heat map button and then be taken back. So now you are seeing this analysis, the arterial hypertension versus healthy living donor. So the one that was associated, not your primary one, but filtered down to just the genes that were from your primary concept, which can be cool. Okay, so that I think is all I wanted to cover in the demo for now, just because I've got a couple other slides I wanted to get by, and I know it's already 1:45. So is there any other things you'd like me to demo for you before I hop away from that portion? Yeah, I hope, and you'll learn later that this is a very small but mighty team. If you have any questions, you can email support, and that's me. So unless I'm on vacation, in which case it's Ross, in which case then it's Rachel. So so please, I'm not scary. I'm just not that smart. So, but if you ask a question, hopefully I can email you back and give you an answer. All right, so let's go back to this. So clicker, standing up, Renly's clapping and winking too, yeah. And then just the third Baratheon king. Here, this is Stannis. So that was a little bit about gene target discovery. And one more person, this is another main character. So we talked about meta-analysis a little. I didn't tell you, but you can actually upload your own gene list. So maybe you developed a gene list somewhere that you think is cool, or you pulled one out of Ingenuity somehow. And you can upload it yourself and use that as your primary concept that you analyze other things against and see if there's anything interesting. And then we did see that you can download things, except for it's mysteriously over there. Okay, so what else are we doing? I said I was really excited to say that now that Nephroseq is back in the university, we actually have some funds behind keeping more than the lights on. I'm proud to say that just yesterday, Ross shipped a release to the app that has a support site, which means that not only do you get me, but I don't get you as often, because now I was able to put some FAQs on a website that we can update, and that's lovely for someone like me. And there was some minor bug fixes. We've got another bug fix release that we're going to do next, and I think we identified at least one today that that we'll probably look at for that. And we can also, anytime we want to make improvements to the data, so the things you saw in that filter tree on the very left, we have to do a release of the whole application to do that right now as well. So there are some data changes we want to make, and a lot of times we try to group them together with an app release. So we'll probably bundle some data changes in. We go drumming looking for new data sets all the time. There isn't a lot coming out right now, but what's happening is that most everyone is switched and they're trying to work on RNA-seq data, and they haven't published it yet. And so right now it's a little bit of famine, but it's going to be feast for us soon. And I'll talk about that in a second. Because that means we have to figure out how to change the app to make sense in RNA-seq, which is something that's imminent and coming for us, but you'll actually see it in a challenges slide right
So
away.
I see,
Yeah.
I see that you say fall, but you mean fall 2016?
I do.
Oh, okay.
Yeah.
Okay.
Yeah, we actually we were able to. I think the technical term was shove some RNA-seq data into Nephroseq right now that we have on an internal version. Chris and Ross did that a couple weeks ago, but we mostly did that as just an exercise in showing all the places where we would need to figure out how to actually make this app work. So it's more than a round peg in a square hole. This was like, I don't know, taking like a redwood tree and trying to like put it through.
It's more three-dimensional.
Okay, yeah, through the color purple. Okay, yeah. All right. So if we get new data though, we want to load it, and we would load it at any time and do a small release to get that data in. We also though think, and we'll get to this challenge here. Actually, oops, hold on, easy Steck. So we're recognizing, especially with RNA-seq data coming in, there's lots of reasons why maybe we should update this app that was originally designed for oncomine cancer researchers 10 years ago, right? And so we do want to do that, but hold that thought. Okay, back up for a second. That's a teaser for 2 slides later. Okay, so let me tell you a little bit just about how this was developed. So Chris, you may have attended Chris's tech talk a couple months ago, weeks ago, on good software development practices. Chris is one of the developers who used to work on Oncomine, and now, you know, Ross is our full-time developer, and we get about 20% of Chris's time right now for Nephroseq. So a lot of this will resonate with you if you were at his tech talk. We follow the Agile software development methodology, which in a nutshell is trying to do lots of little quick releases as opposed to waiting to do a big kahuna at the end. Because a lot of times you end up waiting longer and longer and longer, and the big kahuna gets less and less sexy as you go on. So we try to do frequent small releases, and then get feedback from users about whether that worked or not before, and get that feedback into the next cycle. We have version control. I know that Nephroseq was an anomaly in Chris's like stats about automated tests. And when we can, it's a little difficult with only 20% of Chris and 100% of Ross, but we do pair programming, which is 2 different software developers at one machine, which helps with problem solving. It also helps with knowledge transfer, I'd say. We are trying to get better at moving towards a continuous delivery model, which in software lingo is kind of just something where you can easily ship updates. I don't know if you guys know this, but Amazon and Facebook, they update their app many times a day, and you don't see it. You don't get like a big email that says, woo, we released 4.7.4.2.1.300, because they just do it so often. Now, we're probably not, we've got a developer right now, right? But we want to get to some of the practices that in essence make releases more seamless than they are right now. So that's something we're actively working on right now. And I'll say the pragmatic feature prioritization. So that, as I said, yesterday's release was a support site, right? That's because I'm the support person, but I'm also the person who gets to pick what features come up. And I'm also the person who has to work on a couple other projects. And so for me to just be manning the help desk all day long is not a good use of my time if we can get, nip some of those questions at the bud before someone has to actually email me. And let's be honest, how many times have you actually emailed support versus just like stopped using the app, complained, and then moved on, right? So like we try to get as much help in there. So that's an example of like, that was a feature we decided that was really important to do right away. And we kind of have to make some decisions about what features we prioritize based on the team that we have and what else we're working on. So, that idea of someday re-envisioning the app is beautiful. And I think lots of people are like, yeah, they can get jazzed by that. But you got to get Ross lots more pizza for that to work, okay? Because that's a big endeavor, and we got one dude. So, and for the non-Game of Throners, this is Sirio Farrell. He's a water dancer and a swords master who trains Arya Stark. Rest in peace, Sirio. Okay, all right. so challenges that we're facing. So one thing, and this kind of is nice about the pragmatic feature prioritization, we have a variety of users and then other people like me that we have to always balance and try to think about the best, how do we best serve that population? For instance, we have a lot of computational biologists and systems biologists just within our lab that have very different opinions on what we should do with these data sets and how they'd like to use the application. We have a lot of bioinformatics folks in the lab, and then we have the rest of the world, right? And they have very different views on what we should do. An interesting, actually, a slide ahead here, I want to show you that the Oncomine, part of their software development methodology ended up with them making these personas, so these fake users, right? That they were trying to envision what type of people want to use Oncomine. And it's a really common software development practice to develop these personas and then ask yourself as you're thinking about a new feature, like, is this going to be helpful for Klaus, or is this confusing for Klaus? But before that, you have to pick which one of these people you're actually designing for. And it's hard. In a lot of cases, with Oncomine, and still today with Nephroseq, we can't always decide which one of these people we're doing. Just so that you guys don't have a slide with no Game of Thrones here. I thought the computational biologist would appreciate being the Night King, but maybe I got that wrong. So that is a difficult thing for us, is to try to figure out what decisions to make about new features, because a lot of times it's going to be helping one person to the detriment of somebody else. Another challenge we have is that the world of software has changed dramatically since the days where we had to support lots of users in IE6, right? And a lot of times we'll have really great new ideas come from folks that are used to software that works really elegantly. And we say, yeah, that is a great idea. And I know it's super easy now. It's not that easy for us because, and then we have this big long list of all the complicated reasons why. And we have a lot of new features where, frankly, we're going to have to get better at this. And so this is difficult because a lot of times I use the example of, when do you stop putting money in the used car and just get another one, right? You always kind of say, like, okay, that's my last, I'm not getting new tires. Oh, and then the brakes go out and you're like, what do I do? Do I buy the new car? That's kind of where we are every day with this. A new feature will come up and we'll say, should we try to develop this in the old version of Nephroseq, or when are we going to cut the cord and just start over? That's kind of hard. Oh, whoops, I got these in the wrong order. Especially, this is a great example of one, RNA-seq, right? Winter is coming, RNA-seq is coming. There's no way around that for us. But it doesn't really go well side by side with microarray data in that app that you just looked at. But I bet you parts of what we do in Nephroseq make a lot of sense for RNA-seq data, right? And the idea of being able to draw parallels across data sets, that makes a lot of sense. That's something we want to do. But what do we need to change? Right? Maybe you don't want to look at it on the gene level anymore. That doesn't make sense to roll it all the way up to a gene. But that's what we do all over the place there. So this is a little plug to just say, hey, if you're passionate about this and you have ideas, please tell us, talk to us about them, because this is what we're facing. And you saw summer/fall 2016 is what I meant there. We want to get something out to users this year. And again, in an Agile world, it's okay if it's a very minimum viable product, but we want to get something into users' hands so we can actually figure out, is it the right direction or not? And we're struggling with that. We're also struggling because it's such a small team. So, right, this is only so many things we can do at once. And it's really difficult to get velocity, which is a nice plug for we are hiring. And if any of you are graduating soon, this could be you. Except for probably nobody wants to be Cersei Lannister, so I should have probably got a different piece of image for that. All right. So just wanted to give a quick, let you know who this small team is right now. We all picked who we would love to be, except for Rachel has never seen Game of Thrones, so we picked for her. But I think Todd is a really great person for Rachel. So Rachel's the data curator. She actually works part-time. She works 20 hours a week, and she curates Nephroseq data and data for our Transmart instances at the Kretzer lab. But she's the one who's doing all of that normalization of sample data and setting up the automated analyses to run. She's the one talking to authors, asking for more information, and doing the hard work of slogging through that raw data to make it clean, that it can fit through our pipeline. Ross, Joaquin Hagar, we were practicing this yesterday, is our lone software engineer right now, though Chris, who not surprisingly picked Dario Naharis as his alter ego, gives us about 20% of his time right now. And then that's me, Davos, the Onion Knight. He's a really good guy. Yeah, he did get some fingers cut off once, but I'm proud to say that all these people are still alive as of the end of season 5 here. So you'll have to catch on Sunday at the season premiere of season 6 to find out if they're still alive later. But we did try to pick people that were still living. Okay, with that, no slide presentation would be complete without acknowledging all the other people that helped us get there. Though, as George R.R. Martin, the author of Game of Thrones, tells us, sometimes it's better to not know what goes into this stuff. But obviously, the Kretzler Lab is a huge resource, and they're kind of associated with the George O'Brien Kidney Translational Core Center, which is a lot of letters. And that's also somehow affiliated with the ASBC. Bioinformatics Core, Rich, yeah, thank you for your generous contributions by hiring Chris and Ross to help get Nephroseq something that can still live another day. And then obviously all of that wouldn't be there without the Compendia of Biosciences. So going back to Dan Rhodes from the Wayback Machine, these are the producers of the Game of Thrones TV show. So with that, any last minute questions, or do you guys just want to go get the dessert? Rich.
So I appreciate that the funding is Matthias's and Brian, which is all nephrology, but you know, we got a cancer application, now we got a kidney disease application, there's lots of other phenotypes. Is there any reason we can't do this for diabetes, or you know, pick a disease?
I've held my tongue for a second. Yes and no. Obviously, we've proven that the software can transfer, but then on the other side of that same coin, it's 10 years old, and probably you would want to maybe revisit it. But this concept of taking lots of disparate genomic data and putting it together and trying to kind of make it so it's square pegs or round pegs, that's a lovely concept that I think could work in lots of different disease areas that are, you know, genomic. Have, you know, genomic causation, I guess. And yeah, this would be a great, you know, and I think you would have lots of people that would know what pitfalls we've seen and how to make it work. I think that this is a really powerful application, and I know the folks in nephrology that use it agree, and Oncomine is still going strong now too. So I think that there is a use case there for doing this type of thing with the data. Like there is a persona, right, that wants to see the information at this level, that doesn't want to go deep dive into a sample, right, that wants to look for these parallels across the board. And if we can support those users, I think we should. So, Marcy.
Yeah, actually, I have a question that sort of goes along with that in a little way, but because you guys have concepts in there, have you guys talked at all to Maureen Sartor about linking it somehow with concept gen or LR path in terms of, so then someone might start with a nephrology concept, but then see what other concepts or diseases might be related to?
We have not yet, but that's actually kind of how, remember how I said, full disclosure, the concepts are a little dusty. That is something that's on my roadmap for this summer, is to kind of start banging, I don't remember the phrase, banging doors, knocking on doors. There's probably a different one that I was thinking of, but knocking on doors and asking, what should the concepts that we have in here be? Like some of them are just out of date, but other ones aren't even applicable, and other ones we've never had and we should add. So I will make sure that she's one of the people I talk to, because I didn't know to talk to her. We're going to talk to her about RNA-seq data in a week or two, but I didn't have her on my list for that one. So, yeah, thanks. Anyone else? Yes.
I guess I've got kind of a follow-on to that.
Uh-oh.
And that is, so you mentioned early on that you have used both Transmart and this.
Mm-hmm.
And at a high level, there's a lot of similarities, search, analysis, whatever. And
right.
There's also concepts in Transmart. Is there any crosstalk between those?
There's no crosstalk right now, except that because right now the Transmart uses for the nephrology lab are specifically for the certain longitudinal study consortiums that are using it, and they can see their data set and their data set only, and they can interrogate that. So, for instance, the Neptune consortium uses Transmart, and a subset of the Neptune data is also in Nephroseq. But there isn't any, like, you can't click on, if you're a Nephroseq user in Transmart, you can't click on a special thing that sends you over to one of the other. No integration that way, but some overlap. At least the story I'm trying to construct is that these are that Transmart in some cases is really good for generating new hypotheses because you can do whatever analysis you want, right? But in Nephroseq, you're limited to the pre-computed analyses that we've given you, and so there's sort of different uses that, depending on the question you want to ask, one app might be a little better than the other. So, yeah. But just mostly it's because of the way we use Transmarts, only for this really small user community that has very specific, like even privacy situation, like that we have to keep the data very under lock and key, and it's a little complicated. But I think if I just stand out of the haze of Transmart overall, I think would be a really interesting way. It would be interesting to find out if there were ways to integrate Transmart in general, Transmart corporate, what would you call it? The foundation, yeah, with Nephroseq. Thank you guys for your attention, and yeah, watch HBO this Sunday. Or don't.